My name is Mark Rosenzweig, and I'm a patient like the rest of you. I'm in my 12th year. I'm from West Bloomfield, Michigan, and I welcome everybody to another ALK Talk. We've had over 60 ALK Talks since 2021. This is our third time that we've actually been in an international setting. We were with Dr. Ben Solomon in Australia. Dr. Kilari came in from Italy, and Dr. Katayama is coming to us from Japan. So I'm really, really fortunate and lucky to have people like our doctors who jump in on Sundays. And actually, it's Monday morning for Dr. Katayama. So I'm going to welcome everybody. So I'm going to go ahead and pass over to Summer, who's going to tell us about our future ALK Talks also.
Yes, yes. Well, I'll make you all stick around for that. So, welcome to ALK Talk. It is our third ALK Talk this year. I am Summer Farman, diagnosed in the summer of 2020, and just celebrated 4 years on Brigatinib. I'm Mark's sidekick, and we are so happy to have you all with us tonight. At the end, at the conclusion of the talk, we will give you our sneak peek into our next 3 ALK Talks. You'll have a week off, and then we have 3 talks back to back. So stick around because we will shed some light into what is coming next. But starting his day with us, we are so fortunate to have Dr. Katayama all the way from Japan. He is the chief of the Division of Experimental Chemotherapy at the Japanese Foundation for Cancer Research. He has dove into figuring out targeted therapy resistance. Why are we progressing? Why are we developing resistance? The million-dollar question that we all want to know. So that's just one of his areas of expertise, and he will be talking about that with us this evening. So this morning, we are so happy to have him here. Dr. Katayama, thank you so much, and take it away.
Okay, thank you very much for a kind introduction. So let me share my slide. Okay. Can you see my slide? Okay. So first of all, I would like to appreciate this great opportunity to present our research and also our kind of my history of research on ALK. So today I made this title, The Great Mentors and Friends Through the Research to Cure ALK-positive non-small cell lung cancer. This is because I started this research in 2010 when I was in Boston, MGH. So at that time, I was not good at speaking English also, but my many good friends and great mentors supported, and I could initiate the research on ALK. And since then, I have been working on ALK even now. So that today I would like to talk a little bit history and recent research also. Okay, just in case, I'd like to show my COI disclosure. Even though I received several research grants, but I would like to not, I can say that I'd like to talk this in the neutral position. Okay. So this is my belonging institute, JFCR, Japanese Foundation for Cancer Research. It's established in 1908. It's actually about over 100 years ago. And this hospital is one of the oldest cancer specialist hospital in Japan. Over 700 beds and over 2,000 outpatients every day. And I am belonging to the Cancer Chemotherapy Center and doing the basic and the translational cancer research. So we have 3 research institute, but mainly working on the basic research or clinical research or focusing on the precision medicine. And I am not the physician, but a pharmacist in Japan. I graduated the University of Tokyo in the pharmaceutical department, and I performed the cancer research and got a PhD in 2006 at the University of Tokyo. And then I start working in the current institute JFCR as a staff scientist. And during that time, I had the opportunity to study in United States as a postdoc. And after I come back here again, I now being the principal investigator since 2017. So this is my labo member. Total 31 people are working and doing the research on cancer. And 5 researchers and 6 research technicians and 15 students from the many universities. For example, in the University of Tokyo or Kyushu University or Tokyo Medical Dental University. And also we have several visiting researchers from the company and also from the hospital. So how I got involved in ALK research. As I mentioned, in 2010, I had the opportunity to study in MGH. And MGH is located in the center of the Boston. And I belong to the Jeff Engelman's laboratory in Mass General Hospital. And he was my boss. And so you can see the many postdoc and from the Italy or Japan or some other countries that also been in his laboratory. Okay, and also the Dr. Alice Shaw also was also my co-mentor with Jeff. And every week, Alice and Jeff give 1 hour to have a discussion about ALK. And also the Aaron Hatta, currently he's in charge. He's like after Jeff Engelman left, he is the principal investigator in MGH. And at that time, we were the collaborator. And in 2010, which was 3 years later after Professor Mano's group found the ALK in lung cancer. And as all of you know, lung cancer, especially in Japan, EGFR is the most frequently observed, and the KRAS is the second frequent, and ALK is the third. About 2 to 5% of the patients have ALK-positive lung cancer. In 2007, Dr. Mano's group found the EML4 ALK from the lung cancer patient. And as you may know, that this ALK were not expressed in the normal tissue, but once the chromosomal rearrangement occur, EML4 ALK causes the constitutive activation, and that induced the tumorigenesis. So since these ALK-positive tumors are depending on the EML4 ALK, so that we need to inhibit the ALK by the inhibitors. And currently, many drugs have been developed, and in Japan, these 5 drugs are approved. And in other countries, 6 drugs are approved. And in 2010, the phase 1 study of the Gilteritinib published in New England Journal of Medicine. And that year is year I initiated the postdoc in MGH. And as you can see here, Dr. Alice Shaw and Dr. Jeff Engelman were also the co-author in this paper. And actually, in MGH, many patients were coming to receive the Gilteritinib for as a clinical trial. And I just right after I joined his laboratory, I start studying the crizotinib resistance mechanism. And multiple biopsy sample and prior effusion are collected and used for our analysis. And the patient were very, kindly provide us for the research purpose these materials. And we try to establish the cell lines and also put into the mouse, the immune deficient mice, and make a mouse model harboring the human ALK-positive tumor. And we analyze the multiple mechanisms. And what we want to know is since the molecular target drug works really well, and it lasts usually several years, but eventually the tumor relapse like this CT scan shows. So that we want to know how the cancer survive under the TKI pressure. As you can see here, even though the very nice response were observed, the small fraction of the tumor cells survive. And also how the cancer cell acquire the resistance to TKI. So we want to know how we can also overcome the resistance. So that 10 years ago, or almost 12 years ago, we were working on what is the mechanism of the resistance to crizotinib. And in the laboratory, we treat the cells from the ALK-positive patient-derived cells with the crizotinib. And we experimentally established the resistant cells. And this graph shows that x-axis is a drug concentration, y is the cell survival. So this curve means the red line, the parental cells, 1 micromolar of the crizotinib kills almost all the cells. But after established the resistant cells, crizotinib resistant, we call CR, these cells do not care about the drug, even at the 1 micromolar of the concentration. And 1 micromolar of the crizotinib is about approximately the plasma concentration of the human who received the crizotinib treatment. So we analyze the mechanism, and what we found is one point mutation, only single mutation in the ALK. We call it the L1196M. This is a famous gatekeeper mutation. And we found the very tiny peak, green line, in these cells. So we wanted to report this finding, but Dr. Mano's group, for us, unfortunately, but it's good things for the patient, but they reported this gatekeeper mutation in New England Journal of Medicine in 2010. I don't remember. I do not forget about that day. It's October, and they published, and we were already found this mutation, but we couldn't publish. So that since we collaborate with Ariad pharmaceutical company, which developed the brigatinib, and now that they are in the Takeda pharmaceutical company, but at that time, the Ariad and Jeff's laboratory collaborate together, so that we try to apply this drug to the gatekeeper mutation harboring cells. And the green line and the red line show the similar curve, and even the 100 nanomole, very low concentration, the growth of the gatekeeper mutant harboring cells killed. So that means the brigatinib work and can overcome the resistance. So that we could publish early next year in the PRONAS. And we also studied the mechanism of the crizotinib resistance using the multiple patient-derived samples. So this NZ is actually from Dr. Ben Solomons from Australia. I think because we named the NZ is probably the patient from the New Zealand. But anyway, so that we received the samples from all over the country, meaning in the United States. and analyzed the mechanism of the resistance. And we found the multiple mutations, including the very famous the G1202R mutation. And also we found the some of the case have a EML4ALK amplification. Amplification means that EML4ALK expression extremely become high, and the drug cannot completely suppress the EML4ALK in the but that is only observed in the crizotinib resistance. But anyway, so we found this mutation-driven resistance, and also the other pathway like EGFR or c-KIT or some other pathway-mediated resistance. And before left in 2012, before I left the United States, I could wrap up another paper as a crizotinib resistance mechanism. The one is a EML4ALK amplification, many ALK fusions express. And one is a secondary mutation, G1202R or a gatekeeper mutation. And the other is EGFR or some other pathways activate the cell growth pathway. This ALK or PI3K is like a cell growth pathway. So the EML4ALK usually activates this pathway, and that induces proliferation of the cancer cells. And this other bypass pathway activates this downstream. So after that, I go back to Japan, and again, I start working in JFCR. But Dr. Alice Shaw and Dr. Jeff Engelman kindly accept to do the collaborate with us. And also that I had a great opportunity to work with the physicians in our institute, Dr. Makoto Nishio in thoracic oncology department. So that we started the collaboration in our institute and also our relationship. And this example is our the resistance mechanism in our institute. So we collected also the biopsy samples or pleural fusion and analyzed the mechanisms, resistance mechanism after crizotinib or alectinib or ceritinib treated patient. And as you can see here, after the crizotinib, the many type of the resistance mutation, many variety of the resistance mutation observed. But after alectinib, mainly the two kind of the mutation, and after ceritinib, also mainly the two type of the resistance mutation observed. But other mechanism, the c-MET, which is activating the bypass pathway, or drug transporter. Drug transporter means drugs are once it penetrates into the cells, this pump out by this called the P-glyc protein, and this pump out the extracellular from the intracellular to extracellular, the drug. And along the time, the patient are able to receive the many type of the various ALK inhibitors since the alectinib approved and the Lorlatinib approved. So I was actually feel amazing. The patient can, even the stage 4 cancer, lung cancer, the patient can survive over like 5 years or some of the patient or 10 years. That is amazing because when I start the research on cancer, the lung cancer patients usually survive only 1 year or a couple of years. But now it's more than 5 years. And the patient receives these sequential therapies. And sometimes the tumor relapse, and then we can find the mutation. For example, in this case, in the liver metastasis site, we found the famous 1171 mutation, which is resistant to alectinib. But after Lorlatinib, these tumors were nicely controlled. But after 2 years, another tumors come out. And in this case, in this patient, this compound, like a double mutations, emerge. And in Mass General Hospital, Dr. Yoda, who was in the Jeff Engelman's laboratory and also the Aaron Hatter's laboratory, and now he's in the company. And he reported that approximately half of the Lorlatinib resistant patient have this kind of diverse multiple resistance mutation, like a compound mutation, double mutation, or triple mutation, like here. And actually, Dr. Yoda is my high school classmate. And this is like 1997 in Tokyo. We were in the same high school and actually in the same class. And he became the physician and I became the researchers, basic researchers. And he became the thoracic oncologist in Japan. And he also performed the research. And then one day that he told me he want to study abroad. And I introduced him to the Dr. Jeff Engelman. And once they met, Jeff decided to accept him in his laboratory. And so that we collaborate like after almost 20 years later, after we graduate high school, we could collaborate. But anyway, so that he published this type of the compound mutation. And we also see that this compound mutation. And Dr. Alice Shaw previously reported a very interesting case that after the Lorlatinib resistance, this 1178 mutation, specifically this mutation, sensitize the cells to crizotinib. So in their paper, they showed the crizotinib have an increased affinity by this point mutation. But anyway, so the approved ALK-TKI is effective to some of this compound mutation, but most of this compound mutation were resistant to all the approved drug. So that we try to looking for the drug which can overcome this compound mutation. What we perform is we create the model cells using the mouse cells called the BAF3 cells. And we put in this EML4ALK or this compound mutation. And red is EML4ALK cells. So as you can see here, the x-axis is the cell viability at the 50 nanomolar, very low concentration of the ALK inhibitors. The red, as you can see, the red dot, ALKtNIB or Lorlatinib, that killed almost all the EML4ALK positive cells. But this compound mutation positive cells, the ALKtNIB and Lorlatinib cannot inhibit their growth. But what we found is one of the inhibitor called Gilteritinib shows a very nice effect against the wild type EML4ALK and also the compound mutation, almost like all the cells gone, only the 50 nanomolar of the drug treatment. The structure of the Gilteritinib like this, and this drug has been approved for the treatment of the FLT3 positive AML. And as you can see in this New England Journal of Medicine paper, this Gilteritinib is very active against the FLT3 mutated AML compared to the salvage chemotherapy. So that this is already, I would say, the widely used in the some fraction of the AML positive cancers. And we also tested the activity against the AML positive, like FLT3 positive cells. So we collaborate with a physician in the Nihon University in Japan. And he's also my friends, and he kindly provides these cells. And when we treat the Gilteritinib, these cells died around 10 nanomolar. So the gilteritinib works very well around this concentration. And please remember this concentration. Okay. And for the ALK, we first test the activity of the gilteritinib to the ALK So this experiment is a little bit you may not familiar. This experiment that we use the purified the kinase of the ALK, and we only mix the ALK kinase and the inhibitor gilteritinib, and also the like a substrate. And once the gilteritinib inhibit, the fluorescence will decrease. So since these inhibitors are ATP competitive inhibitors, so that when we use the higher concentration, like 1 millimolar ATP, the gilteritinib require like 100 nanomolar gilteritinib required to inhibit the ALK. And these are 100 micromolar and 10 micromolar. So this curve suggests how the potently inhibits the gilteritinib. And the right is showing the comparing the crizotinib, lorlatinib, and gilteritinib. The point is lorlatinib and gilteritinib have a similar potency against the ALK kinase inhibition. Okay. And another point is gilteritinib have a, I would say, the similar structure with the brigatinib. And as you can see here, that this structure is very similar. That's slightly different, but here different, and here different. And this phosphine oxide also different. But a binding mode, this is predicted by the supercomputer, but this binding mode also predicted as similar with brigatinib. Since the previous data and also this modeling suggests that gilteritinib directly inhibits the ALK kinase. And then we tested the activity against the cells harboring the EML-ALK. And these cells, the EML-ALK positive non-cancer cells, and this one is blood cancer cells have a ALK fusion, NPN-ALK. And this x-axis shows the IC50 value. IC50 value is a required concentration which inhibit the cell growth, the 50% of the cell growth. And please look at this lorlatinib and gilteritinib required almost same concentration as gilteritinib. That suggests that lorlatinib and gilteritinib are almost a similar potency against the wild type ALK. And please look at here that this one is like immunoblot data. The immunoblot means that detect the activating state of the ALK, phosphorylated ALK. For example, the lorlatinib 10 nanomolar treatment completely inhibit the ALK activity, and lorlatinib and also gilteritinib inhibit. And lorlatinib cannot inhibit this double mutant at all, but the gilteritinib can inhibit even the 30 nanomolar. And in vivo data also suggest that C122 is the EML-ALK wild type harboring cells, and this also same, the EML-ALK positive cells. And the gilteritinib can inhibit and as similar as alectinib. So the gilteritinib works very well around lower than 10 nanomolar range, like a 10 nanomolar range, and also that can nicely inhibit the growth of the tumors. And this data is showing the xenograft models suggesting that this mutant, double mutant harboring tumors were resistant to alectinib and lorlatinib, but when we switch to the gilteritinib, these tumors again shrunk by the treatment of gilteritinib. And if we treat the gilteritinib in the beginning of the experiment, the tumors were almost completely gone. So that these data suggest the gilteritinib will work for the treatment of the compound mutation positive, some kinds of the compound mutation positive cancers. And this slide shows the IC50 color plot, and green is active. Active means the drug can inhibit, and yellow is probably be able to inhibit, and red is probably resistant, and black is resistant. And for example, the crizotinib have many, these are single point mutation, and these are double mutation. And how we can look at, for example, the 1171N mutation, resistant to crizotinib and alectinib, but sensitive to brigatinib, ceritinib, and probably lorlatinib. And gilteritinib works really well against these single mutation, except for G1202R mutation. And also, this compound mutation, many of these compound mutations are resistant to lorlatinib, but sensitive to gilteritinib. So from this data, not all mutant, but some of these compound mutant might be able to overcome by the gilteritinib. I really appreciate for the initiating the clinical trial of gilteritinib for ALK-positive patient by the Dr. Angela Tin in Michigan University. And I heard that this is because of the 7.6 million gift from the Julie Stern Foundation. And I hope that this trial also starts in Japan, but not at this moment. They are not initiated yet. Okay, so from here, I a little bit talk about the more like a basic research things. We are considering why most of the case the tumors are not completely gone, and they hide and then eventually relapse. And also that in some cases that we need to consider the intrinsic resistance who do not well respond to the initial treatment. And when we look at the clinical trial result, for example, in this crizotinib, the first phase 3 clinical trial result, most of the patients, the tumors shrunk more than 30%, but a few, several patients were start growth immediately and not shrunk well. And also if we look at 30% of the patient, even the alectinib, the 20% of patient relapse within 6 months. And if we look at 1 year, about 30% patient relapse earlier. So what is the difference between this early relapse and the late relapse? So we want to know the mechanism and how we can target these tumors. And if we look at the alectinib, brigatinib, and lorlatinib, the situation are similar. Within 12 months, the 20 to 30% relapse within 12 months. So among the ALK-positive patients, the 20 to 30% of patients cannot receive the benefit of the drug. So that we need to understand clarify the mechanism of those initial poor response. So in Japan, we have been working on understand what is the mechanism of the resistance, especially I am focusing on the early resistance. And we called ALKIWA study, the real-world observational study. This one was supported by the Chugai pharmaceutical company because we only use alectinib first-line treated patient. And we collected the 250 patients in Japan. And first cohort A is already received the first-line therapy, and cohort B is who initiated the alectinib after enrollment. And we monitor the tumors, and also we collected the blood every 3 months for the cohort B, and also we collected the blood in the when the tumor relapse, especially in the cohort A. We only collected the resistance tumor or blood sample, and of course the cohort B also. And we also collected the diagnostic slide, the FFP slide who diagnosed, patient diagnosed. And using that tiny specimen, we perform the sequencing and check the email for ALK variant. And the frequency were similar. And we now almost complete analysis of for the patient. And this data is a second interim analysis. We are now analyzing the final result. And this is the PFS data of all the population. The left is a cohort A, and the right is a cohort B. And the left shows the PFS is 102 months, very, very long. And cohort B is 28 months. But this is very consistent with the previous clinical trial data. And the reason why the cohort A showed very long response is when we enroll the patient already received already 2 year or average 2 year of the ALK treatment, which suggests the early relapsed patients were not included. So that also strongly suggests that once this early relapsed duration, once the patient passed this duration, probably the tumors are nicely controlled and the PFS tends to be very long. And as you can see here, within 12 months, the 30% of the patient relapse. But after that, the declining rate were very mild. So we really need to understand this mechanism. Today, I'm not going to talk about the detail about the finding about this, but in the near future, we will be able to publish this mechanism. And when the tumor relapsed after the ALK treatment, we analyzed the mechanism of the resistance using the blood samples. Since the blood samples have some limitation about the detection rate, so that we could not detect all the case, but about 20% of the patients that we detect the mutations, which is almost all the well-known mutation as a ALK resistance mutation. And another scientifically interesting point is PFS. In the cohort A, the mutation detected patient were relatively shorter progression-free survival. So that suggests the mutation positive cells already exist in the earlier time point, like when the tumor diagnosed, patient diagnosed as ALK-positive cancers. So that we need to more clarify the mechanism why this phenomenon occur. But anyway, so the patient will tend to receive the very longer treatment of the ALK, but unfortunately, some patients relapse. And we are now also analyzing the second-line treatment. After the second-line treatment, what is the mechanism of the resistance? Okay, so we are now trying why the TKI, tyrosine kinase inhibitor, cannot cure advanced cancers. So that we are now focusing on the drug-tolerant persistent cells. In the many literature, in the many paper that people publish the mechanism of the resistance of the drug-tolerant persistent cells. And since we can use the clinical samples, so for example, if we inoculate the G12 mutant tumor, we can analyze the like almost all tumor gone, but we can analyze this tiny tumor. For example, if we treat the G12 tumor with the Lorlatinib or Brigatinib, we can see the remaining tiny tumor. And we were looking for how they survive. Okay, so since the already timed, I would like to finish my talk here. And at the end, I'd like to thank to all the patients and also the families to provide the clinical samples, and also the lab members who give me chance to study ALK with me. So that I'd like to thank to all the members in our laboratory. Thank you very much.
Thank you, Dr. Katayama. Fantastic, so many great points. For our ALK community, this is such a great reminder that we only know about ALK and lung cancer because of the Japanese. Right? In 2007,
yeah,
they found the ALK gene, the ALK fusion in lung cancer, and that's why you're alive. So I think we're grateful. Second thing that's really important is that Dr. Katayama and other researchers in Japan are publishing more data on ALK all the time. So this is not just one person with 31 people in his lab. It's gigantic. Third, we wouldn't have the Gilteritinib trial if it wasn't for the work that came out of Dr. Katayama's lab. And I know some of you aren't familiar with Gilteritinib. Gilteritinib clinical trial at the University of Michigan is funded by you through ALK-positive. We are paying for that. We're also, the board is expanding that trial to additional centers. The maker of Gilteritinib is a pharmaceutical company called Astellas. And we, from the medical committee of ALK-positive, have been on the phone on multiple occasions with Astellas, asking them to expand the trial to Europe and to Japan. And we seem to be getting some enthusiasm for that. So Dr. Katayama, hopefully your high school buddy, Dr. Yoda, will have something else. But if you think about what we're trying to do as an organization, one of those is to figure out why TKIs don't cure people. And people like Dr. Katayama are focusing their entire career on that. Gilteritinib is one of the first fruits of that work. And there are other things that we're working on. And lastly, I'd like to just so you know, so Gilteritinib is approved for acute myelogenous leukemia. So far, they've treated 4 patients at the University of Michigan, and the toxicities seen in leukemia aren't showing up nearly the same in ALK-positive patients. And that's primarily because the leukemic patients have had a lot of prior chemotherapy, which makes their bone marrow reserve and other things, makes them more vulnerable. So we're encouraged very much by the results so far in the Gilteritinib trial. So thank you, Dr. Katayama and your colleagues. I also wanted to comment on the ALK-cure trial. By the way, I love the name, ALK-cure. That's great. And when I read the publication, it was very impressive that people were doing so well. And I wanted you to know that part of that, as Dr. Katayama mentioned, part of that is the design of the study. You see that really long benefit because they didn't enroll patients till they'd been already 2 years on ALKtALK. So that meant the patients who weren't going to respond to ALKtALK weren't enrolled in ALK-cure. So it basically meant that you had what Dr. Kamage would call the good ALKs, or what are enrolled in the ALK-cure trial. But I'm hoping the ALK-cure trial is going to really give us a lot more information about resistance. And so that will benefit everyone. So thank you again, Dr. Katayama and your colleagues for that. Finally, Dr. Katayama and his colleagues are working on the other resistance mechanisms. In December, I had the privilege of going to the Hot Topics in conference in Washington, D.C., where I met Dr. Katayama in person. And many researchers were there talking about how we're going to figure out the resistance mechanisms to TKIs, not just ALK, EGFR, ROS1, and so forth. And I think one of the things that I've learned from Dr. Katayama and the others is the development of resistance to TKI is complicated. It is complicated, and there are multiple things going on at the same time. And as one person mentioned in the chat, some of it might already have been there, but the drug that you take allows it to show itself. And some of it, of course, is being caused. Some of it is because you're getting the drug, the tumor is adapting and developing resistance to it. And that's very complicated. So as sort of his title was about great mentors and friends, it takes all of our research community working together to solve something this complicated. And I was very encouraged by the meeting in Washington, D.C. in December that there's a good collaboration. And thank you for highlighting that in your presentation. I really appreciate that. So I'd like to now just see, Summer, there were, of the different questions, were there some that you'd like to start with for Dr. Katayama?
Let me go through. Ken, that was an excellent summation. And Dr. Katayama, this might have gone over our heads, but just knowing all of the diligence and the team and the collaboration and the thinking outside the box to keep us in the game, we are all, if you look at the chat, we are all so very grateful. So on behalf of everyone, which at the end we'll all unmute and thank you, but you know, our deepest gratitude. We are so grateful for, as I like to say, what happens while we are sleeping. So we don't care that it's over our heads. That's what we have Ken for, to help bring it to us. And it's amazing to know what is going on. One thing I did want to point out, that we asked that question in the beginning about what if you had progression, what did you do next? And that was kind of a trick question from us too, because we can't stress enough. And as you saw, the different ways you can progress is you can't really go after the beast until you know what's driving it. So the more information you can get, the better. It is not just a sequential TKI to TKI. You need those biopsies, especially tissue, if you can get it. Blood is great because it's easy, but nothing helps reveal what is driving the cancer like a tissue biopsy. So someone had asked questions about that in there that I saw. Let me go back and see. And Ken,
I have a question, Summer, while you're going back through.
Oh.
So Dr. Katayama, I've asked you and some of your colleagues, and there doesn't appear to be an ALK patient group in Japan like there is here in the United States. So one, is that true? And two, how can we as a patient group help you progress as quickly as possible the work you're doing?
Thank you very much. Thank you very much. In Japan, unfortunately, I don't think that we have a group of the ALK-positive patients. But I may a little bit miss, but we have a group of the lung cancer patient, like a group, but not the ALK-positive patient. I think it might be cultural things. I'm not sure. I'm not fun. I don't know. But for example, if the patient is like an infant or a child disease, we are more have like a patient group that easily to get because the parents actively work. But in Japan, the patients are not so actually work to make this kind of like your community. So that is a situation in Japan. And how the second question is how patient group the support. I actually appreciate this kind of like Gilteritinib trials go forward by the ALK-positive funding and also the foundation support. So that is a big needed strategy because sometimes the pharmaceutical company cannot make decision to start the trial. So even though that it might have some potential, and we don't know that before we test, but if the patient groups can get together and push us forward to doing, so that is very strong pressure. And also another thing is this kind of like opportunity, if we present the biopsy samples used for this purpose and make models, and we can analyze and detail. So if you understand the importance, the patient are more positive to give us the sample, fresh samples, even though that it takes time and it takes some pain, but it's really needed for the future development and future finding. So it's sometimes the reluctant to take the samples. It made a little bit difficult, but it's really important for us to do the more research. And one more thing is it's kind of a good pressure for us. We need to do the more and find it more, because since I'm a researcher, but I tend to go into the too detail. But important thing is what occur in patient, so that we need to realize what is the important things, what need to identify, and we need to sometimes understand not only new finding, but also the needed finding. Do you make sense? Okay.
That's beautifully said. Thank you.
Yes, that all makes so much sense, actually. And that was something that I was practically screaming about at TTLC recently, was that it's so exciting with the crown data, and that is fabulous, but that's mostly fabulous for people just starting out that are newly diagnosed. Most of us here tonight all need these options. So the need is there. Just because we have these TKIs, that doesn't mean, eventually we run out of those options. So we're so glad that you're still continuing to look for other options. And you bring up a great point with the tissue samples, and if you're being biopsied anyway for progression, perhaps then some of that could go to the University of Michigan where they can compare. So that is a really good point, and we can discuss that further. But also, one of the questions from the chat was regarding brain penetration, or even more specifically, leptomeningeal disease. What are your thoughts on that, Gilteritinib helping to combat that?
Thank you very much for question, important question. To be honest, I have no experimental data to show the activity, the brain metastasized tumor. But I guess probably that it's going to not small fraction of the drug going to the brain, and also in the brain metastasized tumor, the blood-brain barriers were usually destroyed. So even though it's not going to the actually in the brain, but in the tumor sites it may leak. So the answer is I don't know. So I actually need some data.
Yeah, so we'll need more research in that, and we'll see how that works out. And for people looking to put this option in your back pocket, right now it is just at the University of Michigan, so you could go on clinicaltrials.gov, or you can reach out, I'm sure in the Facebook support group or through ALK-positive. We could definitely help you make connections. with the University of Michigan. And like Ken told you too, we are hoping that they will expand to different sites as well. And you can learn more about that actually when Ken gives a medical committee update on April 6th. So we'll have a medical committee update and you can find out what is happening behind the scenes at ALK-positive, what are our medical committees doing and what projects are they prioritizing? What are we doing about persister cells? Because they seem to be a real problem for us. So we need to get rid of them. So you can find out more about that on April 6th. I don't know, Mark, did you see, were there any more questions?
I think there's a question there about resistant mechanism. When you take Lorlatinib, it seems that you develop these double compounded mutations. And when you presented your chart with the heat map, and it shows that Gilteritinib does do compound mutations. So do you feel that, I know I don't want to do the sequencing, but Lorlatinib is for people who progress on alectinib typically or brigatinib. Do you think that Gilteritinib could be a different choice after Lorlatinib, or do you think that's a drug typically before that, before it?
Thank you very much for the question. It's a difficult question, so that I answer is I actually don't know. I have no idea about that. Which is better. So yeah. So and also I'm not a physician, so that
I know you're not a clinician, so
Yeah,
it's hard to
yeah.
tell that.
yeah. Sorry. So
So I'm sure that that's something that we won't really know because there's not going to be a study upon that. But it's kind of interesting when you show that map, and I'm sure Ken can describe it a little bit better than I can when you show that heat map there of what explains to other people what that map represents.
The heat map that Mark is referring to shows that relatively which mutants are killed by which of the drugs, and in the laboratory. And this, to me, Mark, the most important thing about that heat map is you need to be being treated at a center who's used to taking care of ALK patients, used to using all these drugs, used to looking at this data. It's not simple. And I think it probably warrants a second opinion in very many cases, because to figuring out which drug, which followed by which drug for which mutations, what if there are no mutations, it's very complicated. So the heat map, many of the ALK specialists that I've spoken to don't use the heat map anymore for patient select, for deciding how they're going to treat a patient. In part because they're using Lorlatinib in first line almost all the time. And then what is approved for post Lorlatinib? Nothing. So the whole sort of paradigm of treatment is becoming more and more complicated. So I encourage everyone to make sure that if you progress and your site can't do a biopsy or won't do a biopsy, get a second opinion and make sure that's not just the skill level of the people at that institute or experience of the people at the institution. Because without the tissue, you aren't going to be able to decipher all these mechanisms. And as Dr. Katayama showed in the ALK-cure trial, the sensitivity of the blood assay is limited. So yes, do the blood, but you'd rather, knowledge is power. And an experienced oncologist who knows how to use that knowledge is the best.
And I'm sure we'll
I don't know if that added anything, Mark.
not, well, I mean, it's a complicated question, but
It is really
you know,
hard.
right. I just want to stress that, this is a worldwide disease, and obviously Ken and the medical committees are bridging everyone together for us. And I know Jeff Sturm was involved in reading papers from Dr. Kanamani and trying to hook up the drug company with his research to get us to this point. So it's really taken everyone's energy and efforts to do this. So I really appreciate it. And no, I just think that it's all interesting research, and we really don't know how it's going to work on us until many years later. But it's very interesting. And Ken, I don't know if you have any closing comments before we wrap this up.
No, I'm so appreciative. Dr. Katayama, thank you again I've been really looking forward to this.
And thank you.
Yes, this was amazing. We did have questions pop in about the side effects, and I think it's so hard to tell with only having 4 patients been on it, and we know that the side effects that ALK patients experienced are far less than the patients that were battling leukemia faced, but we don't know specifically. So time will tell, and we can find out more information for you. Also, people were asking if you're in a country where the trial isn't available, can you get the drug off label since can it be prescribed since it's available for leukemia?
Yeah, so we've met with a doctor from France, and he was able to get it for patients there through their health system. So that's where we have to get out and advocate for yourself and for the community in whatever country you're in.
Okay, sorry guys, I think I froze over here.
Yeah, you froze on us.
Sorry, everybody. Thank you for taking.
Well, Dr. Katayama, go enjoy your day. It's 9 o'clock your time on Monday. Thank you so much for coming in and joining us.
Thank you so much.
Let me just give you the preview so you all can mark your calendars. You have a week off. And then on March 30th, so in 2 weeks, we have Dr. Catherine Wu from Dana-Farber. She is a medical oncologist who specializes in stem cell and cell therapies. So she will be talking to us about the roles of cell therapies and the importance of immune response in treating ALK cancer. And then I gave you the sneak peek that on April 6th, we have Dr. Ken Culver, our ALK-positive director of research, coming on to tell us what's been happening with the medical committees. We're so grateful for all of the funds that we have raised collectively. Now, what are we doing with that? So he will talk to us about that. And then there are a lot of questions and concerns regarding chemotherapy. What is chemotherapy? Some people use it up front while they're waiting for their ALK diagnosis. Some people are using it as a systemic consolidated therapy after they've been on a drug. Some people are keeping it in their back pocket as a reset or as a last resort. So we have Dr. Shirish Gagiel coming on, and also a few patients who have been on chemotherapy to talk to us more about the role of chemotherapy in treating our cancer. So look forward to that. Also tomorrow is the Death Cafe with Dana. So join her at, I believe Dana is at 1 o'clock tomorrow Eastern time.
Yeah, 1 o'clock Eastern time on the same Zoom channel.
Yep, so you can join in to that to talk about the role of accepting help in your cancer diagnosis. Do you like it? Do you not? You want to shove people away? Or maybe you need a little bit of support. And then also, if you are interested in joining a private roundtable, which is not on our calendar, just a group that will meet twice a month from April until July, and then possibly on and on, please email ALKtALK@ALKpositive.com. So that is it. Thank you all for joining us. Oh, .org. Okay, don't email .com because we won't get it. If you would like to unmute, please unmute and thank Dr. Katayama.
Thank you, Dr.
Thank you very much.
Thank you, Dr.
Thank you.
Thank
Thank you so much for the hard work. So appreciated.
Thank you.